Good morning, everyone. Shwetikanta Guho Foundation is a charitable society which was established in September 1991 in memory of Shwetikanta Guho, who expired in January that year. Since the names of Shwetikanta Guho and his wife, Preetilata Guho, the first secretary of the foundation, are indissolubly linked with South Point as its founder and its associate founder, the first members of the foundation decided that education should be the main sphere of the foundation's activities. Currently, Shwetikanta Guho Foundation runs 4 schools in West Bengal. Garden High School, Kolkata, affiliated to the Council for the Indian School Certificate Examinations, Garden Eye International School, IGCSE, Kolkata, Garden High School, ISER, Kolkata, affiliated to the Central Board of Secondary Education, and Garden High School, Shonarpur. By setting up the Shwetikanta Guho Center for Culture and Learning, that is SKGCCL, the foundation aims to nurture young talents in the fields of art and culture. Shwetikanta Guho Center for Culture and Learning conducts a variety of short-term and long-term programs in communicative English, foreign languages, music, dance, drawing, painting, photography, short filmmaking, etc. SKGCCL, in an effort to supplement the formal curriculum offered by educational institutions, organizes cultural programs, webinars, seminars, and workshops, both online and offline, on various subjects and topics on a regular basis to ensure the overall development of young learners. Such programs are open to all educational institutions with specific target age groups for each program or event. This morning, it is my pleasure to introduce our special guest, Dr. Deeppaman Ganguly. Dr. Deeppaman Ganguly is a physician scientist and human immunologist with research interests in basic immunology and autoimmune diseases. He went to Rahara Ramakrishna Mission High School. He did his MBBS from Medical College, Kolkata in 2001, PhD in biotechnology from CSIR, Indian Institute of Chemical Biology, Kolkata in 2006, and PhD in immunology from UT MD Anderson Cancer Center, Houston, USA in 2010. After a postdoctoral stint in Columbia University, New York City, USA, he started his laboratory in CSIR, Indian Institute of Chemical Biology in 2013. He is a recipient of Swarna Jayanti Fellowship from Department of Science and Technology in 2017, National Bioscience Award from Department of Biotechnology in 2018, and Shanti Swarup Bhatnagar Award in Medical Sciences 2022. Big round of applause for Dr. Deeppaman Ganguly. We are really thankful to Dr. Ganguly for taking the time out of his busy schedule and to be here with us. Please accept a token of appreciation from us. Dr. Ganguly, if you could please come up. Over to Dr. Ganguly.
Can you hear me? You are not hearing too much, right? Too loud or something? No. Okay. So tell me something. So whatever was being talked about.
Yes. Closer to your mouth.
Okay. Is it fine? Okay. So whatever was being talked about this, about while introducing me, it's very kind of them. It's a very kind gesture that you call me up here. But that's not the point. The point is, I study immunology. I do research in this field of immunology. Now tell me, so one fine morning, if your teachers bring one immunologist in front of you, what kind of questions will you like to ask? Anyone? I mean, let's make it little informal, I mean, otherwise if we don't discuss, it will not be very clear. So anything. So what comes to your mind as a question when you hear this term immunology? Anyone? Anyway, okay, so we are too shy. I thought it's Garden High School. So, I mean, you should, one of the best schools in the state. I mean, my daughter also comes here. So I take pride in your school as well. So keep up to that. Anyway, so whenever we hear about immunology, it's all about this. I think these days after the pandemic, the first thing that comes to mind is the big hoardings with written immune boosters, right? All these advertisements on immune boosting, etc. Isn't it? And even when people are buying baby foods, you find this components, immune boosting in baby foods. But what I'm planning to talk about today is so many things that we actually do not know in immunology. So it will be a talk on ignorance about immunology. That's why it's titled like that, so confusion and excitements. Now, whenever you are ignorant about something, for few things you are confused, right? And there are things when your ignorance and when it is resolved, you are excited. So I talk about two such things. One is an ignorance that we always thought we know about it, and still when we think deeply enough or new data come to our fore, we find out that no, we didn't really know about this properly. So that creates confusion. And there are, as I said, new developments that no one ever thought about, and that brings you the excitement about in the field. And fortunately, I would say fortunately for my students or for me as well, we have been witnessing some of those, we have been part of those confusions and as well as the excitement in this field over past 20 years of my research career and for my students for almost 10 years. So we'll start with, I'll just take this thing. Sorry. Yeah, so all of you know your immune system, right? Some of you have to answer some of the questions, otherwise it will be really bored. You will feel sleepy after some time. Yes. So where in the body the immune system lies in our body? Do you know? Anyone? Where is the immune system? Where is the heart? Where is the liver? Where is the GI tract? Abdominal cavity. Where is the immune system? It's all over. That's the point, right? It's all over. So it's all over. It's all over the body. And that should be there, right? So immune system is giving us immunity. If it's just restricted to one part of the body, how can it give immunity to some other part of the body? So it has to be all over. And honestly, there are some body organs or contraptions made by nature which are really specific for immune system, like lymph nodes, lymphatic vessels. Perhaps some of you have learned about it, very little about it. I don't think in class 10 I knew about lymphatics, but I mean, if you don't know about it, this thing you will find out later on. But all over our body there is this skin boundary. So an organism has to be a bounded state, right? So there has to be a boundary. So the skin is our boundary. And there are internal boundaries, right? So gut lumen, there are walls. So all over those boundaries, immune system is acting because it has to protect us. Now this word, it has to protect us. It went to you very normally. And I will show you, there lies the confusion, whether really it's there to protect us. And if you really want to do research with immunology or research with immune system, where do you find the immune cells? In the blood. Which part of the blood? No. Well, in plasma, yes. I mean, some soluble factors are there. You're very right. So if you take blood from your vein, if I might draw some blood from my vein and put it on a tube and spin it really well, there will be a centrifugal force, so the heavy things will go down. And those heavy things, now some of the heavy things will go down really fast because they are dense. And in blood, the most dense corpuscles are RBCs, red blood cells. They are full of iron-rich protein called hemoglobin, so they will just go to the bottom. And then all the white blood cells come over. So you can actually see white blood cells here, right? So if you do it, then you will find out there is a pack of white blood cells. So all the immune cells lie in this white blood cell compartment. That we have learned in the school, isn't it? But immune system being so complex and working all over the body, it has to have a lot of subsets of cells. This palette of white blood cell, they will have all these different kinds of cells and even more. So there are different kinds of cells. Why there are different kinds of cells? Why do we have different kinds of cells in our body? Anyone? And if you answered this, I will be really elated. But why do we have all different kinds of cells? No, not forget about infections. Just you have your skeletal muscle cells which look different. You have skin cells which look different. Exactly, fantastic. So that there is a division of labor. There is a division of labor in our body. So we are all multicellular organisms, and there is a division of labor. Some cells have taken up some responsibility, others have taken up other responsibilities. Same goes for immune system as well. So there are cells in that bunch of white blood cells which actually take up different responsibilities. We are not going to know about those responsibilities. You will find out when you read biology. How many of you plan to take biology at some point of time? Fantastic. Congratulations to all of you, because next half a century will be of biology. Whoever is still thinking whether to not take biology, reconsider it. Please reconsider, because next half a century, if not the whole century, will be of biology. Why? Because we don't know anything in biology. In physics, in maths, in chemistry, we know a lot. Why? Because it started much earlier. Modern physics, if you take Galileo to be the forefigure of modern physics, that was when? Huh? No, give me a year. Early 17th century. What was happening in early 17th century in India? Akbar was there. Akbar was in Delhi, right? Isn't it? Akbar was in Delhi. That's when physics started. Modern physics. If modern chemistry starts with Lavoisier, when was that? What? I can't hear you. Whatever. Again, it's early Mughal dynasty. And if modern biology started with what? Can you tell me? What kick-started modern biology? Not really. I would say, there can be, we can argue, but I would say it started with this voyage of Beagle. Do you know what is Beagle? What is Beagle? That is the Darwin ship, right? So Darwin took a voyage and he came back, he wrote Origin of Species, right? And that was when? That was around 1859. What was happening in India at that time? Sepoy mutiny. So physics, chemistry started when Akbar was on the throne, and biology started when Sepoy mutiny was going on, right? So we are kids, we are kiddos in natural sciences. Naturally, you have plenty of things to discover. We don't know much. We really don't know much. So the upcoming centuries, we are almost 1.5 century behind physics. Naturally, the coming 1.5 century will be ours. We will discover new things. So whoever is not considering biology, just reconsider. So all these cells have their own responsibilities. We are not going into this. And all of these come from the bone marrow. So this is called hematopoiesis, right? Hemato is from the Latin prefix, I think. Yes, I would say Latin. And so that's blood. So blood is being formed from the bone marrow. So right? So all these cells are coming out. These are stem cell, hematopoietic stem cells. Perhaps you have heard in blood cancer patients, what you do is you replace the hematopoietic stem cells. Because there is cancerous blood cells, so you replace the hematopoietic stem cells, the new hematopoietic stem cell, new blood will come up, cancer will be gone, right? So this is where all the immune cells start forming. Then I'll start talking about the confusion I was mentioning. So we think it protects us against everything. So that's why they are present in the barrier. That's how we started our discussion, right? So what is the foremost barrier? This is the skin. Well, skin is, we think that it's a very thin thing, but it has a lot of things, right? So there are epidermis, dermis, etc. It's not important. Details are not important. And in the dermis itself, there are a whole lot of immune cells. And it's always doing a surveillance all over the body, right? So this is a normal skin section. Can we just switch off this many lights, some of these lights? Is it here? No. I think it's there. Yeah, I think it's fine. Okay. So as I said, so this is I think it's behind this board, so it will be difficult. Yeah. So as I said, so this section, there is the dermis and all these immune cells are there, they are playing their parts. And whenever there is a breach of infection, a wound, so people were playing, I saw, they were playing soccer on the field, so some of them will get some bruise, and then there will be some, that's bound to happen, right? That's the whole point of playing a game on the field. So there will be bruises and your immune system will take care within 2 days, right? So that's what it does. So there is a breach in the epidermis or something, all these immune cells come over, they start producing a whole lot of chemicals that will excite other immune cells, inform other immune cells, okay, we have a problem here, just come over, do something together and find health, right? So let us get back the health. So that's how they play a role in all the barrier surfaces. And all these cells can be found in this blood, this circulating blood, white blood cell blood. And do you know these people? Very interesting people. Both of them are very interesting people. So he found, while working with sheep blood and pig blood, mouse blood, etc., he found there are specific chemicals in the blood that can react to specific toxins introduced into the blood. So, and his name was Paul Ehrlich. Paul Ehrlich, perhaps you'll know afterwards. So Paul Ehrlich is also famous in medical science because he discovered first chemotherapeutic drug. So that was salvarsan. That was used to treat syphilis. There was no chemical drug before that, okay. Antibiotics, that's stunning, right? But this is a chemotherapeutic drug that will be used for treating a specific disease. Do you know what is the second chemotherapeutic drug that was discovered? And that came from this city. Uriastilamine, Kalazar. Who did that? UN Brahmachari. That's the second chemotherapeutic drug that was discovered all over the world. That was from this city. And so he comes from my college also. So he actually studied medicine in medical college. So we take great pride. And so Paul Ehrlich first showed that there are specific chemicals that can be formed by the body that reacts against specific chemicals. Now we know these are called antigens, antibodies, whatever. But the specificity is very important, right? So that means the body is adapting itself to the challenge it is posing against. Okay? So this is called the adaptive way of doing immune response. But this Russian guy, Eli Mechnikov, he was looking at something else. He was looking at starfish larvae and poking them with some bulbs, some plant bulbs. And he was finding under microscope that there are cells coming around those wooden bulbs and reacting against it. And he was actually looking at an event called phagocytosis. That means if there is a foreign thing inside the body, some cells will be there who will go there and who will try to engulf it and digest it. So that was, and that is called an innate response. Now these two guys, they were not very friendly to each other, naturally, because they were all discovering really key things in the immune system, and they knew of each other. They knew that they are competing against each other. Ehrlich thought that the immune system always works like that, engulfing things and digesting things. And Mechnikov and Ehrlich always thought that it's all about the specific chemical reactions. Nevertheless, they got Nobel Prize together. That's the irony. And they were both given Nobel Prizes in 1908 together to usher in the age of modern immunology or mechanisms of immune system. Now, all this fight was actually ingrained in the flow of evolution. I'll tell you why. They were fighting against each other, but they were actually looking at different systems. So Ehrlich was looking at mammals, rats, pigs, sheep, etc. And this guy was looking at starfish. Now we know that in the evolution of organisms, of the animals, so this is the unicellular life. And we'll come back to this. Well, there lies a great confusion. So it is actually related to the confusion. Unicellular life, then comes all the primitive multicellular organisms like sponge, corals. They don't have any bilateral symmetry. For example, I have bilateral symmetry, right? Two of my hands are similar looking. Two eyes are similar looking. There is a midline. So we have bilateral symmetry. Now, primitive organisms didn't have any bilateral symmetry. They had almost like tree, no symmetry. And then came starfish, etc., whatever. Point is, this adaptive immune system, where the immune system will actually recognize something specific in terms of chemistry and react against it, came here. That was after this jawless fishes were being developed, vertebrates were developed. So the specific immune response is only present in vertebrates. Now this poor guy, Paul Ehrlich, was looking at vertebrates, so he was always finding specific immune response. And Mechnikov was looking at starfish, so he had no idea about specificity because there was no specific immune system. In that, I mean, they didn't know about this. They thought that every animal will work similarly, but no, they are on the evolutionary line. So that's the origin of their conflict, right? So that's why he discovered specific immune response and Mechnikov discovered innate response, phagocytosis, very primitive type of immune response. Now the point is, okay, so immune system, there is specific response, there is non-specific response. Even if you give a particle of wood, that will be phagocytosed, engulfed, it will try to digest. Why all these things? Just think about so many different cells, right? So it's millions of cells in our body, billions of cells in our body are dedicated to this function. Why? That requires energy, that requires ATP, a lot of respiration, lot of glucose, right? All has to be channelized to keep this thing going, and it is working all around, I mean, all your life. Why nature will invest that much of energy to do that? That's very important. In biology, when you think about biology, any problem, if you do not think about evolution as the kind of justification platform, you are not really thinking. So you have to always think that how do you justify anything happening in any biological system through the path of evolution, through the history of evolution. So naturally that's a big question. So naturally the question comes, why do we need immune system? Now in 1950s or late 1940s, there was someone called Robert Venn who was doing some experiments, and he was doing experiments with pigs and also mice. What was he doing? So he was trying to see whether you can transplant skin from one animal to another. So initial days of graft rejection, transplantation science, right? And he was trying to do that. And it was getting rejected. If you take skin from one mouse, put it on another mouse, it's like debride the skin and put the skin on another mouse, it's getting rejected. And then he found that if you introduce the recipient mouse with some cells of the donor when the recipient is too young, they are very small pups. Now when you are introducing some cells from the future donor into that recipient when it is still a pup, and then it becomes adult, and then you come back with a skin graft, it actually accepts it. That gave this idea to the whole immunology community that okay, so that means this selfness, what is part of me and what is not part of me, it is defined early in life. And if early in life someone sees something, or the immune cells see something in the body, they will take it as our self. And as the organism grows old, then it is fixed, then it cannot be changed, and so this acceptance happens. And that was the major. So all these theories come from one or other experiments, right, and the data. And then people try to give a general model so that the general model will explain the data as well as make sense. So based on those data, these three guys, so this is Peter Medawar, he worked in London. This is Frank McFarlane Barnett, he worked in Melbourne, and Joshua Lederberg worked in USA. Now, Peter and Frank were really good friends. They were working together. They were always writing letters to each other and discussing their own thoughts. And Joshua mostly worked by himself. But anyway, so all of them actually suggested that the immune system is there just to make this self-non-self discrimination. That was the first time people actually asked this question, why do we have immune system? And this self is defined early in life. So that was a model given by them. So this is for immunologists, for all of us, it's called self-non-self model of immune system. Now this was accepted, and on the school books also, how do you get introduced to immune system? Immune system comprised of immune cells which protect us against pathogens, whatever, right? So that totally goes with the self-non-self model that was given by these three people. They also got Nobel Prize for giving this theory in 1950s. I think 1956 or something. So they said that the major question that the immune system is asking, which part of the body is me and which part of the body is not me. So that's the major question we are trying to ask, right? And that's the whole function is all about. Now let us go back to I already talked about one part of the body called skin, which is a major area where immune systems are always fighting because that's exposed to the environment. Always we are having wounds and bacteria and viruses, whatnot. So this is the skin of forearm of an unfortunate individual. You can see there are kind of plaques forming, shiny silvery plaques. Have you ever seen this disease? This is a disease. Huh? No, not eczema. It's called psoriasis. Perhaps some of you have, right? Psoriasis is a very common disease. Very common disease. There is no infection, nothing. One fine morning, a young guy or a young girl discovers her skin is full of these plaques, and plaques are forming, and these lesions widen, and it actually remains for all over life. When it's on the skin, it's not that bothersome, but in a lot of patients, it actually affects our joints and bones, and that becomes really difficult, very detrimental for well-being. Now here there is the infection, and so this is the skin as it looks like in this disease of psoriasis. The epidermis has grown inside, and there are like finger-like projections, and the epidermis is getting thickened, and when it is getting thickened and proliferating and cells are coming out, that are coming out as those plaques, these silvery plaques, because it's keratin-laden cells. That's why it looks like a silver thing. And it's an autoimmune disease. There is no infection, nothing, right? So the skin is being acted upon by the immune cells as if there had been a great assault by a foreign organism. But there is nothing. But still, the immune system, a lot of immune cells are flocking in there. And this pianist, this guy, his name is Michel Gillier. He used to play jazz. He also had 3-4 jazz albums. I happened to be a second PhD student of this guy back in Houston. So he was a doctor, he was a dermatologist, but left clinics and came to research. And I actually did PhD with this guy. He's from Switzerland, and he's a Swiss guy, but we worked together in Houston in the Anderson Cancer Center. So what Michel showed, along with his students and postdocs and the whole team, the team of Michel showed that there is a very intricate immune response happening in the skin in these psoriatic individuals. Especially what he's famous for showing is that self-nucleic acids, DNA molecules that are present in the skin cells or in our all body cells, they are being acted upon by the immune cells. Immune cells are recognizing our self-nucleic acid, self-DNA, self-RNA molecules as antigens and acting upon that. And that's how the disease starts. And that's a big question, right? So it actually questions the whole point of self-non-self model of immune system, because it's acting against us. So it cannot really distinguish between self and non-self. Then why is it there? Also in cancer, so this is a cancer cell, right? And this is a lymphocyte called T cell which kills cancer cells. And you will be surprised to know this thing is happening all over the body all the time. So all of us, from our very childhood, there has been cells which tried to be cancerous. all over our life, immune system is protecting us. That's the truth. So there are micro cancers or micro tumors forming all over our body. How do we know that? So people have done very interesting experiments. They have taken animals, they have got rid of their immune system, they just kept them as to get old. And when they're getting old, they're full of tumors. That means all over the body it's happening, because some cells are, I'll talk about the evolutionary term of this, we call it cheating. So some cells are always trying to cheat the system and grow on its own, and immune system is taking care of that. In fact, these cells at times can evade the immune response. Perhaps some of you have read in the newspaper, they can actually evade the immune response, and this harmonica player from New York rock band discovered some molecules so that this evasion can be stopped. His name is James Ellison. So he developed immune checkpoint therapies. Have you heard of it? I think in the newspaper also it comes, right? So all these cancer immunotherapies. So all these cancer immunotherapies was developed by this harmonica player. So what Jim showed, that there are molecules that can be targeted when the T cells are being evaded by the cancer cells. You can actually stop this evasion, and he got Nobel Prize for this, developing cancer immunotherapy in 2018. So he also now works in MD Anderson in Houston. So I already showed you two contexts where immune system is actually trying to fight against self-entities. Cancer cell is part of our body. It's one of our cells that is proliferating, but immune system is acting against it. The skin is part of our body, it's acting against it, right? And again, this was something that was first cooked in our lab. Now a whole lot of other labs are following it up. And so all these awards that you heard about was because of this. So a lot of obese individuals, they become diabetic, right? And people know that in their, they have an inflammatory state. Have you heard of the name inflammation? In biology of heart, right? So there can be inflammation on the skin, which is all these classical signs, and also inflammation inside. And so we actually went to the surgeon and looked into their fat which is present in the abdomen, that's why they are obese, and looked into them, and we found that a whole lot of immune cells are there. So Amrit actually did that long back. It was 2015, 15-ish. So now he is in MIT Raleigh Institute doing his own postdoc. So he found out that in the fat there are a whole lot of immune cells which are actually getting activated. Now what are they reacting against? Here is a guy, a poor guy who loves to eat. He ate good food, got obese. What has immune system to do with that? Why are they reacting? That's amazing, right? So you don't know why they are reacting. And forget about Medawar, Barnett's self-non-self model. Here there is no self-non-self conflict. Here only conflict is whether to go for ice cream or biryani. That's it. But it's happening. So that leads to the confusion. Why exactly we are having immune system? We don't know why we are having immune system. That's why I told you, biology is rich with questions. We don't know so many things. We assume we know. We don't know. If you take a deeper delve into the contemplation, you will find that you don't know. Now this question was bothering a whole lot of people. This lady, Polly Metzinger, amazing immunologist and philosopher. So she first proposed back in 1994. I think her model is the most influential model. I think I'm doing well with time, right? Is it okay? Yeah. So I think she has been phenomenal. So I mean, this is not the place to discuss about it. I mean, she has been phenomenal in different way. So she left science after doing graduation, was a waitress in a bar or something in California, There some professors used to come and she used to talk to them, and these professors found out that, well, this lady is really smart and she knows biology, and they kind of dragged her back to the graduate school, and then she did PhD at the age of 40, and then she became Polly Metzinger. Okay, so she is one of the greatest philosophers in immunology and also experimentalist. So she proposed that immune system senses danger. It doesn't have to be a self or non-self encounter. Immune system doesn't care whether they are looking at a bacteria, virus, or your self cell. It only cares about whether it's going to create any danger to the system. Well, I'm not going into that, there are controversies against, this model has also been questioned, but by far this is the most stable model in the immunology since 1994, since Polly proposed it. It's called danger model of immune system. So the immune system is there just to sense danger in the body. But the quest is not yet off. There is another guy, much later in 2013, he proposed another model for understanding why we have immune system. So you can just understand, we are starting from, immune system is immunology is an amazing subject where the application came first before we really knew about the science. Edward Jenner developed vaccines long back. They didn't know anything about immunology. Still we don't know a lot of things about immunology, right? So the application came first. It started saving people much earlier than the science was being developed. And so this guy is again very interesting. How many of you are planning to go into humanities? History, anyone? History? Philosophy? Philosophy, anyone? Okay, so this is your friend. He's a philosopher. So he did his philosophy major from Sorbonne, Paris. He did his PhD in history of science, rather history of immunology. And he proposed the most recent model of immune system. So what he thinks, Thomas Pradu, I had the opportunity, I didn't meet Polly Metzinger, I have talked to her over email, but this guy I actually met in person, and we are in touch. So Thomas says that immune system senses change. If there is any change, it starts reacting. It may not be danger as well. Perhaps his model fits more with the obesity thing. So there is something is changing. The body doesn't yet know whether it will be, because, well, obesity leads to cardiovascular disease. That's 20 years down the line. But immune system is working right now. It doesn't have that future vision. But it's responding to change. And that's what Thomas proposes, that's how immune system works. And that's why the system is there to sense change in the body, something like that. Now let us be a little bit more philosophical, just to show our camaraderie with your friends who are going to take philosophy. Go ahead, philosophy is great, fantastic. And if you study philosophy, think of science, philosophy of science little bit, come back to science and help us. Because scientists are really bad philosophers, and we need philosophers like Thomas Pradu in the field. Now the question is, do you know what is me? For example, someone comes from outside, an alien comes, and I am standing here like this, an alien appears here. And he looks at me, I am moving arms, etc., talking something, making sounds. He will think, okay, maybe this is an earthling. Yes, this is an earthling. But what will be his first impression about the earthling? How does one earthling look? So he will think an earthling looks like one free limb, another something attached to a big block of thing, isn't it? So he will think that this podium is also part of my body, right? So he has just appeared now. He has never seen me, how a human being looks like. So he will then figure out when I come out of that, okay, so he can come out of that and he can move out of that, that means it's not part of his body. But he cannot shed his arm down the floor. So that means the arm is part of his body. Isn't it? That means it's very difficult to define me. Me means individuality. What is an individual organism? It's very difficult to define. So evolutionary biologists have tried very hard and they could come up with these definitions. That means an individual is countable, non-separable, because this is separable. I could get separated from this. That's why it's not part of my body. And I can be counted. Mr. Mark can be counted, right? Something like that. And there is a trans-temporal identity. That means after half an hour also they will find that these limbs are part of my body. That's it. That's how the organism can be defined as individual. Otherwise everything has questions. Philosophical hook, biological hook, and there are other questions. And now you think, if this is individuality, now you think how it came into being through evolution. We all came from this evolution, right? So this is the evolutionary tree. Sorry for this very small thing. So here sun ignites 5 billion years ago. Earth forms 4.8 billion years ago. Moon forms around 4.4 billion years ago. First prokaryotes come over 4.2 billion years ago. Not 3.8 billion years ago. Okay. And the sponge and fungi, they come when? 800 million years ago. So the unicellular life comes at 3.8 billion years ago. And multicellular life comes at 0.8 billion years ago. Evolution, unit of natural selection, survival of the fittest, everything is applying. That means on this planet, unicellularity or one cell was the unit of natural selection. For how long? Almost 3 billion years. And then suddenly multicellularity was developed in different parts separately, is fine. Now a whole bunch of cells come together and they relinquish their right to be the fittest. Now they say, well, individual cell doesn't have to be fittest. Now this collection of cell has to be fittest. That means there was a shift of individuality. Did you understand this? Anyone who didn't understand, just raise your hand. This is very important. In biology, this is very important. Okay, I'll repeat it. If you can just take one message from this talk, this should be it. So, for the time on this planet, life was there. 4/5 of the time, life was unicellular. 1/5 of the time, life was multicellular. That means, for those 4/5 part of the life, like almost 3 billion years as I calculated, the evolution was happening on single cell. Right? All the survival of the fittest. So unit of natural selection was single cell. And suddenly with multicellularity, the unit of natural selection changed. It became multicellular. And there came possibility of cheating. Because now we already talked about, right, while we were talking about cancer. So there is always a possibility of cheating, because now there is a collection of cells which relinquishes their own claim for fitness, own claim for becoming a unit of natural selection, and they now become a collective, and they think this collective should be now selected through the process of evolution. And there will always be cells. But just before that, in their genome, in their genes, the compendium of genes they have all the genes that can let them live independently. Still they are relinquishing this right. Now there will always be chance that some cells will try to cheat out of this collective system. And some of us think that's why the role of immune system comes. That's why, so there have been very interesting experiments that were too difficult for you. That's why I decided not to talk about it. There have been very interesting experiments where an organism is unicellular and multicellular in its own life within 24 hours. Some slugs, some Shaula, do you know this Pupuro Shaula slugs, Dictyostelium organism, and they have shown that when they go from unicellularity to multicellularity, the first division of labor, that's why I talked about division of labor. The first division of labor is of sentinels, is of immune system. The first special function that comes in some of the cells is phagocytosis, engulfing foreign objects. So it seems, because there is this chance of transformations or oncogenic transformation, hote pare, or other kinds of things are possible. So whenever there is multicellularity, there will be chances of some cells going rogue and cheating the system, and that will create problem, and that's why all this cellular immune system is there. It has to drive, this is just I said. So why would a cell follow its right to be the fittest, and who defines the unit of natural selection, and what if there are cheaters? And so this was first proposed by Stephen Gould, a Harvard evolutionary biologist, that they are bound to be cheaters if there has been unicellular to multicellular transition. And more recently, Athena activists said that there must be immunosurveillance because these cheaters have to be restricted so that multicellularity is possible. So Athena actually proposed this one. So maybe multicellularity was the reason, or maintaining multicellularity is the immune system function. We never thought of it before. But still, it's an ongoing field. As I said, I mean, it's a very ongoing field. Athena is a young biologist in Arizona State University. She's of my age, so she's still working on these things. Now, so it seems perhaps immune system is there just to ensure a conflict-free multicellularity. So the multicellular organisms are devoid of internal conflicts, and that can be, so we don't really know. Now I'll just point to two things that immunologists are really excited about. One thing is, we recently have realized when an organism is more than itself. What does it mean? So it means that in our organism, in our body, we are full of different microbes, right? So all of us have heard of it, gut microbiota. And so for diseases, people are giving probiotics. Why? To give good bacteria into your gut. So people have actually, so all our, not only in the gut, oral cavity, skin, stomach, it's full of different organisms. And they have been there. We are realizing they are there today. That doesn't mean that they were not there when our first predecessors went out of Africa. They were there. We never knew about them. That means for last 300 years, Homo sapiens biology has been developing, keeping them inside our gut. And now we know that in our body, if you count the cells, someone did a theoretical calculation, this many cells are of our genome, right, our DNA, and this many are microbes. So our body, is it ours? Not really. Then who is me? We don't know who is me, right? So your genome doesn't define your me, because the number of cells in your body is more coming from the microbes that you harbor in your body. That's amazing, right? More importantly, as I said, so they are actually playing very interesting role in our physiology. They actually play important roles. Sorry, I think So one student in my lab, I was talking about this immune activation in obese individuals, right? We also did some mouse experiments. I'll just briefly talk about it. So anyway, so some immune activation will drive insulin resistance, type 2 diabetes in a mouse, right? Which we discovered from human beings. Now this is just insulin resistance curve. So red means these mice are insulin resistant. They are not responding to insulin. That means diabetic, plainly. Now what this brilliant student of mine did was, she took, Purvita, so she's just finishing up, she took feces from these mice, made very nice, delicious poop shakes, took wild type mouse and made them drink it, okay, those poop shakes. So some group of mice were having poop shake from healthy mouse, and some mice were getting poop shake from this immune activated mouse. They were not being, their immune system was not being activated. They were just being given poop shake from mice whose immune system was activated. And now see, just having poop shake from these immune activated mice made them diabetic. There had been very interesting experiments. People have made even delicious poop shakes from the feces of Parkinson's patients. Took animals, made them drink it. They were having neuronal problems. Okay? Can you realize? So we don't know anything. We are still scratching on the surface. That means these many cells that are more than in our body, they are actually playing very important role in our physiology. We never knew about it. We are now knowing about it. Okay? That's excitement. It's exciting, right? It's very exciting. And sorry. Now I'll end with this other thing. So what do you think? Immune cells are talking to each other, immune cells are doing something in our body. How do you do that? What kind of information they are processing? What do you think? Mostly chemicals, right? That's how we have thought about biology. That's why in the joint entrance examination, people ask you to read biology and chemistry to become doctors. I hope in 50 years it will not be like that. I'll show you why. So we think that biological system or all the immune cells talk to each other through chemistry. Now, that's also very fantastic. I mean, perhaps you have known about. So this is what is this? Cell membrane. What is the model? Anyone? Fluid mosaic model. Yes, perfect. Singer-Nicholson's fluid mosaic model, right? So this is lipid layer. This is a lipid bilayer, and there are proteins on the studded in there, like a mosaic. So that's why it's a fluid mosaic model. And usually the biochemicals that take part, this is serum albumin, a very common protein. So they are like fitting into each other. All this chemistry is happening, and that's why they're recognizing things. So there will be chemical mediators released by one cell. There will be another chemical sitting on the surface of another cell. They will talk to each other. They will bind to each other like this, and that's how all these activation, all these functions come in. So it's very well known that yes, much of the biological system or biological cells talk to each other through chemistry or biochemical mediators. And that's becoming really exciting after artificial intelligence came in, because all these modelings can be done now on artificial intelligence platforms through machine learning. Perhaps some of you have known, AlphaFold has come from Google's desk, so which actually creates all these protein structures based on machine learning. That's also exciting, but that's not the excitement I'm talking about. I'm talking about this stuff. This is a very interesting protein. I think it's the best looking protein or best functional protein in the body made by nature. It's called piezo. Piezo word comes from pressure, Greek pressure, right? And this guy discovered it, Arden Patapoutian, back in 2010, just 13 years back. So he works in Scripps. And he actually went on to win a Nobel Prize but for discovering it within 13 years. Just within 13 years. That's amazing. Usually people get old, they almost die, then they get Nobel Prize. And this guy got Nobel Prize when he was like 49. Just got it because he discovered this channel. And now, why did he get Nobel Prize for discovering a protein? Now look at this figure. This is how the protein works. It's a very interesting looking protein. It's on the cell membrane. This is the cell membrane. You said the fluid mosaic model, this is a fluid. And there are two blades, blade-like structures of the protein which are jutting out of the membrane. And when you pull it, protein sidewise, the blade-like structures come down, and a central pore opens. And then some ions like calcium or sodium goes in. That's it. Nothing involved. That means this is a Galilean lever in action. This is a protein, the function is just Galilean lever. A blade comes down on the pool, central pole opens, ions go in, cells are activated. That's it. And how come this can happen? So this is a membrane stretch. That means cells also sense mechanical cues, not only chemical mediators. We never thought of it before. We never thought of it before. And interestingly, a very talented student in my lab, Chinky Shuchen Liu, Chinky is from Kolkata, by the way, don't get confused by the name. She is from Kolkata, she went to, I think, Carmel, and St. Xavier's, etc. So what Chinky Chinky did, so this is this lymphocyte talking to another cell. It can be a tumor cell, it is not in this case, it is tumor cells. So it comes here. So just think about it. It's a fluid mosaic membrane. Two cells come together, push to each other, and then there will be a lot of stretch. So now we know cells can actually sense this stretch and make use of it in their operations. And I'll show you, so what Chinky asked in the question, so cells have to migrate, right? So in the blood, these cells are there. Perhaps you have known, during inflammation, cells will come out of the blood, go to the tissue. That means they're migrating, they're walking through the matrix, extracellular spaces. And so this is a T cell migration, right? So a T cell is migrating on a culture plate. This is in our experiment. And so when a cell is moving, how do you move? Do you know? How do we walk? You put your first foot forward, then push it through muscle. That's how you propel your body up forward, right? If you cannot do this, you can't walk. That's why you can't walk on an icy surface, because this force is missing. Cells, when they are walking, they also have to do that. So that was the question. And is this channel involved in this force sensing while cells are walking? So usually cells, when they walk, so the leading edge, which is the front foot in our case, so leading edge of the cell, the front of the cell is sensing force, giving force, and propelling the cell forward. And what Chinky did, she took T cells, or lymphocytes for you, and got rid of this gene. This piezo1 was absent from these cells. Now this is a control cell. They are nicely moving, right? So there is a direction to which they should move. They are nicely moving. You can see the tracks behind with a microscopic image. And when you get rid of the cell, get rid of the piezo1 in these cells, cells have lost their ways. They cannot really move in the right direction. And so this is the piezo1 reorienting itself to the leading edge. This is a cell moving. This yellow signal that you see, that's the leading edge piezo1, and the cell is moving forward. So that means, apart from chemistry, physics play a very crucial role in immune functions. And we never had any clue about that. I'm happy that my laboratory was able to contribute in this field as well. So I'll end here. These are the one confusion, great confusion. We are still confused why immune system is there. Perhaps we are inching towards the right answer. And there are plenty of excitements. I just talked about 2 excitements. So whatever you study, biology, philosophy, engineering, mathematics, we have place for all of them. We have nice problems that only you can solve. Study and then come to immunology. Thank you.
Students, if you have any questions, you can just ask now, or else we can also share the questions later on. But if you have any questions, you can ask now.
If there is any burning question, I am happy to. I may not know the answer to that question, but you can always ask. Yeah, please, sure.
Sir, I have one question. You said the single cells were stable and there was no scope of cheating. So why did multicellularity begin?
That's a very, very good question. It's $1 million question. It seems, so there have been again different models, because when multicellularity was being developed, we were not there. So we can only make guesses, make models and then fit with data. So it seems that there were local stringencies in nutrients, right? Local adversities. And then if 5 cells came together, their survival was better than 1 cell being there. So if they could divide their labor of being alive, they could fare enough, I mean, they could fare well. So that's how the locally, in different places, multicellularity started. And that also perhaps is true because multicellularity was being developed at different parts of the earth, but all around 0.8 billion years ago. That means Earth's atmosphere was in a right state for making those stringencies or making the environment too stringent for single cell to be alive in some places on the earth. And that's why it kind of, in the history of planet, it actually falls in a very short window of time when multicellularity started. So it seems the easy answer will be, which is not, we don't know whether that's the right answer, but easy answer will be local need for being together. Otherwise they were not surviving. Was it good? No, not good answer. Yeah, sorry, I know. It's not a good answer. Yes.
Good afternoon, sir. My first question is that you said that there were various philosophers and scientists who have said that immune systems can sense danger or they sense change. My question is, how do they sense that?
That part we know better. So that biochemical pathways, what is it sensing, that we know better. There are different receptors. The fluid mosaic model, there are proteins on the cells. So some of those proteins bind to other chemicals which are released by another cell. And so whenever they sense that their protein is bound to something which should not have been bound in this respect on that context, then they react, then they get activated. So that part is much more charted out, I would say. So when you go to college and study more, you will find out. But the major question lies, why all these things are happening? That was the confusion. And that kind of borders onto philosophy, but it's very important to learn about human health also.
Sir, my final question is that you had said that when a foreign particle is introduced into a person's body at a very young stage, like in the case of the mice, it was introduced at a very young stage, then that particle gets recognized.
What particle? Foreign cell.
One particle gets recognized as as the body's own particle. Then my question is that in case of very young children, when they require certain kinds of transplants or any sort of organ or blood donations, then why do we have to match the proteins and other things? Like, is it too late for it to be recognized?
Very good question. The problem is, in our case, in case of Homo sapiens, that stage is inside our mother's womb. Got your answer, right? So for mice, they are outside for that stage. But our early development, when it happens, we were inside our mother's womb. So we can't be given a transplant when we are, say, 1 day old. We'll react.
Sir, I'll end up with one final question, if you don't mind, which is that you mentioned about cheating. Some cells try to grow on their own. So would you say that AIDS is also a form of cheating? the cells of the AIDS, the virus, they also try to grow on their own?
Well, viruses are a little different compared to this cheating, but I think you got the point rightly. So if you think of this microbiome, which I named, I did not utter the word holobiont, right? So in our body there are different microbes which are part of our body. And if you think of that as the organismal structure, I don't know whether you will understand, but I think you will understand. You have got the point. Now you can actually think of infection as one microorganism which was not part of your body. It wants to be part of your body for some survival advantage. Okay. So that's how different viruses, different bacteria came into our body. And we never reacted because they were not causing any severe danger to our living. So this virus or bacteria or parasite, you can think in this way. This parasite, it comes to your body just to be part of you, just to be part of that holobiome. Most of them are accepted, some of them are rejected. The rejected ones, we call them infections. You understand my point? Perhaps. Yeah, so it almost borders on to philosophy, but I think that's the way to think about it, yes.
Thank you so much, sir.
Anyone else?
Thank you so much. Have fun studying. That's important, having fun. So whatever you study, please have fun, otherwise there is no point. Thanks.
Thank you, Dr. Ganguly, for coming and speaking to the students. I think if you have any questions, you can write to him via us, so we can send in your questions to him and he can respond. So if you have any questions, you can send your questions to me, and I can forward the questions to Dr. Ganguly.